All right, we're going to pick up now with the last of our lectures on the reproduction part of our chapter, and then we'll move here relatively shortly into the chapter, or the rest of the chapter, which is on development of especially animal development. So with fertilization then, what we're going to see is now the actual union of the sperm and egg cells nucleus. This, like we've talked about before, typically occurs in the uterine tubes or the fallopian tubes. Doesn't occur anywhere else, and if it does, there's typically something that's not right, or the pregnancy is not going to move forward if it does occur that way. In most cases, we're going to be talking about now hundreds of sperm cells actually getting to the egg cell, and that's really important because it takes more than one sperm cell to get through the outer layers of the egg cell. So in this picture up here in the upper right, first off, the first thing you notice is how different in size these cells are. Each individual sperm cell is a tiny fraction of the size of the egg cell which they are surrounding. These sperm cells with their acrosomes have to be able to get through the outer layers of the egg cell. So we call those outer layers, in our case at least, corona radiata and the zona pellucida of the egg cell. Once that sperm cell, or once those sperm cells get through there, one of them is going to stimulate the egg cell to go through meiosis 2, finally finish off meiosis 2, and then eventually at that point there is fusion of the two nuclei and a zygote is produced. And so this picture here we can actually see the two layers, the corona radiata and zona pellucida. Now once that one sperm cell penetrates that layer and fuses with the nucleus of the egg cell, there's going to be a very, very rapid chemical change that's going to occur that prevents any other sperm cells from getting through and prevent any further nuclei from fusing together with the egg cell and the pre-existing sperm cell. Because if that were to occur, then you'd be talking about a zygote that has too many sets of chromosomes, and that of course just simply wouldn't work. The last little bit of this chapter deals with fertility and how people can limit fertility, so contraception basically is what we're dealing with here. There are a variety of different categories of contraceptive techniques, ranging from preventing pregnancy in the first place, both permanently as well as temporarily. Right? So you can actually look at a full-fledged sterilization situation where what you're going to do is block the pathway of sperm cells or prevent sperm cells from reaching an egg cell. So in the case of a vasectomy, there's going to be a cutting or severing and then cauterization of the vas deferens, which connects the epididymis up to the urethra, which is right here. If you cut and sever that connection, then sperm cells are still produced. Spermatogenesis continues to occur just like it would normally in the absence of vasectomy, but the sperm cells simply cannot exit the male's body. So of course they can't reach the egg cell, and again you have no fertilization taking place as a result. A tubal ligation is kind of the same idea now, only in a female. We're going to have now the uterine tubes are severed and sealed off, typically with a very small clamp, and that's going to prevent sperm cells coming up from here from encountering egg cells coming down from here. So there's now a blockage in between there so that sperm cells can no longer reach the egg cells. And typically what they'll tell you is if you choose to have either of these procedures done, they should be considered permanent. There are cases where maybe they can be reversed, but in most cases that's a very risky proposition, so they are more or less considered to be permanent means of contraception. As far as temporary conception, a variety of things here. The first one we'll talk about is a chemical prevention of ovulation. So what we're going to be looking at here is, first off, birth control pills in women, which are high doses of estrogen and progesterone. So basically what's happening here is we are taking advantage of the body's negative feedback system by dosing it with estrogen and progesterone, which then prevents LH and FSH from being released from the anterior pituitary. And if those two hormones are not produced, then you're not going to have maturation of a follicle and eventually ovulation from the follicle. By maintaining high levels of estrogen and progesterone throughout most of the menstrual cycle, that's going to prevent ovulation from occurring. Barrier methods would involve things, again, temporary things to help prevent sperm cells from encountering egg cells. So condoms, diaphragms, those kinds of things, interrupt sperm travel, typically also used with some sort of spermicide to physically kill off the sperm cells before fertilization could occur. Probably the best way to get pregnant is to use what's called the rhythm method, which is where a couple would attempt to sort of guess, this is a pretty decent way to say it, guess when ovulation might occur, and then avoid intercourse during those times when the egg is actually present and when it could be fertilized. Now, the reason why I say this is guessing, and the reason why this is many percent less effective than any of the other means, is that it's often very difficult to tell when ovulation might occur, unless you're really committed to doing so. So unless you're committed to monitoring body temperature and monitoring the um, production of certain chemicals during the menstrual cycle, it's very difficult to tell when ovulation might actually occur. The other issue with this is that sperm cells can have upwards of 2 or 3 days worth of a lifespan inside the female's reproductive tract. So even if ovulation hasn't occurred when intercourse is occurring, if ovulation then occurs later, you could still indeed have fertilization. So that's a fairly low efficacy means for preventing pregnancy. Another possibility would be to prevent implantation. So with what's called an IUD, or intrauterine device, this is a device that's implanted in the uterine lining, which helps prevent embryo implantation. And so the idea here is that fertilization might still occur, but if it does occur, then the zygote cannot implant because the uterine environment is not conducive to implantation anymore. There is also, for typically what are considered emergency circumstances, what's called the morning after pill, which is where you administer a really high dose of estrogen and progesterone in one dose to prevent implantation. So if someone is concerned maybe that a pregnancy might occur when it's unwanted, then a morning after pill might be an option in some cases, but certainly not something which you want to rely on for your sole means of birth control. Beyond that, it's a matter of actually physically removing the embryo from the uterus. So you'd be talking about an abortion of some type. So now the actual embryo and placenta would be physically removed from the uterus, or in some cases, not typically, or not in this country, legally at least, there's a chemical called RU486, which was first introduced, I believe, in France, which actually stops progesterone production during the first 7 weeks of pregnancy. And of course, if you don't have progesterone, you cannot maintain the uterine environment, and so menstruation would occur, and with the menstruation would also come the embryo also at that point in time. So it basically induces menstruation after the pregnancy to help remove the embryo from the female's body. So that is the end of the reproduction section of this chapter. So I will wrap up here and then very shortly get started on the development section of this chapter.